Thanks. That's what I get for putting Brian on the spot. Okay. All right. So I get the privilege of introducing our speaker this week. So before I give her formal introduction, we're just so thankful for Dr. Sullivan to be here. As many of you probably noticed throughout the day, Dr. Sullivan had to take 3 flights to get here, and it's going to take her an entire day to get back. And when I give you her introduction, this is going to just show how much more meaningful. She's dean of her graduate school, and she really volunteered her day to us so that we could, all of us had the opportunity to meet her and hear about her research and just get to know her, which is, it's been a fantastic opportunity. So to introduce Dr. Sullivan, she's an internationally recognized expert and leader in the field of sex differences in cardiovascular physiology and pathophysiology. Actually, when the request for seminar speakers came out, unbeknownst to each other, Dr. Padilla and I both asked if we could ask Dr. Sullivan to come. So again, just so fantastic that she's here. Her work's been continuously funded by the NIH and the American Heart Association since becoming her tenure track faculty in 2008. Dr. Sullivan consistently publishes her work in top journals in the field of hypertension with over 100 peer-reviewed articles that have been cited over 24,000 times or 2,400 times. For her outstanding work, she's received numerous prestigious awards, including the inaugural John Lara Research Award from the American Journal of Hypertension, the Mid-Career Award for Research Excellence from the American Heart Association Hypertension Council, and an Outstanding Faculty Member Award from the Graduate School. As further evidence of their status in the scientific community, Dr. Sullivan has active leadership roles in national scientific societies, such as the American Physiological Society, the American Heart Association. She serves on the editorial board and associate editor for leading peer-reviewed journals, and she's a standing member of an NIH grant study section. In addition to all of her scientific achievements, like I said, Dr. Sullivan is an exceptional educator and mentor. She's demonstrated commitment to the training of undergraduate, graduate, and medical students, as well as postdoctoral fellows and junior faculty members. And she's a dean of the graduate school currently at Augusta University. She creates a challenging, collaborating training environment for all of her trainees while focusing on the needs of each individual. And these are not just things that you write for a fantastic bio. I think we can all say, as we've met her today, these are genuinely true. It's just been fantastic. To date, she's successfully membered a number of postdoctoral fellows, PhD students, undergraduate students, and medical students. Her trainees routinely earn prestigious awards, travel awards, research and recognition honors. So she's really dedicated not only to her extramurally funded research, but also to the scientific community and to the success of her trainees. And so we're so thankful that Dr. Sullivan is here, and I will pass it on to you.
Well, thank you very much. I feel wildly unworthy. It's been an absolutely phenomenal day. The opportunity to meet with all of you, talk with all of you, the trainees at lunch, every single interaction I've had has been outstanding. Highly impressed by the collegiality that you all have shown me, the collaborative nature and environment developed here. It's really quite impressive, and I feel very honored to have the opportunity to be here. So you do ask me to do this relatively late in the day, so we'll see how well I can remember anything that I was supposed to say. So the focus of my lab really for pretty much the entire time I've been in Augusta, which is my entire academic career, has been focused on sex differences in blood pressure control. I'm going to give you kind of a couple different stories. Hopefully they make sense coming together. The first part kind of leads into the second part somewhat, but maybe not at all. So you can give me your feedback later. If at any point in time anyone has a question, please go ahead and put up your hand and stop me. I would much rather have a conversation than just talk at you for an hour or an hour and a half. We'll see how long I want to stay up here. So hypertension continues to be a significant clinical problem. More than 1.13 billion people have hypertension, and if left untreated, it leads to a number of different complications across a variety of different organs, including stroke, heart failure, erectile dysfunction, loss of vision, heart attack, kidney disease, and kidney failure. Because of this, there becomes increasing interest in treating hypertension. This is looking at numbers since 1990. And what you're looking at in this figure is those undiagnosed with hypertension. So the big red down here, and this is number of people in the million. So I'm not good at this, but we'll say 400 million people, starting here to about 500 million people have hypertension, but it is not diagnosed. You have those that are diagnosed, and they know hypertension. You can on the brain say, yes, I'm going to lose that pesky 45 to 145 pounds, and that's going to help me decrease my blood pressure. You have those in the light green here who are treated with medications but do not achieve blood pressure control. And then you have the few in the bright green here, and the encouraging piece is the bright green gets larger. Those are the individuals that have hypertension. They are being treated for their hypertension, and their hypertension is controlled. But what you should get from this is that is a lot of people. And if we could effectively remove hypertension as a problem, the number of lives that would be saved is phenomenal. So controlling blood pressure and eliminating hypertension as a complication would really save millions of lives. When you talk about lives, we're talking about obviously both men and women, and you're talking about a variety of different ages. You know, historically, hypertension was thought of as a disease of old men. It is not actually the case. So what this graph is showing here is the prevalence of hypertension in men who are in, let's see, does this work here? Yep. Men in blue and women in red at different age ranges. So starting at the very young, 20 to 44, up to the 75 plus crowd. Two things that I'd like you to note. So one, yes, there are sex differences in hypertension. So in younger individuals, prevalence is higher in men than it is in women. But even in what I'm going to define as young women, about 20% of women have hypertension. So it's not something that is strictly seen in men. It is also seen in women. By the time you reach over 75, more women have hypertension than men do, but really at this point, does it matter? It's like 80% of the population is going to develop hypertension. So hypertension is something that afflicts just about everybody. And if you look at it between the sexes, most people do not know they have it. That big red box at the bottom, they don't know they have hypertension. That's about 1 in 4 men and 1 in 5 women. So yeah, I won't make you raise your hand if you know you don't have hypertension, but having your blood pressure checked regularly and understanding what your blood pressure is a simple thing to do. It is the primary modifiable risk factor for a number of cardiovascular diseases. So having ways to treat hypertension becomes important. This is a slightly different way of looking at that graph I showed you before. So that green bar at the top is an encouraging number as you look at it like that. But if you look at it like this, it's not necessarily as good as we would like it to be. So of the percentage of individuals that are treated for their hypertension and have it controlled, like 28% or 26% of people. So the majority of people that have hypertension are doing things to lower their blood pressure unsuccessfully. So we have lots of treatment options. We know there are lots of different ways to lower blood pressure. It's easy enough to check. It's a not invasive thing to do. We know of the importance of exercise. We know of the importance of eating correctly, especially the audience I'm talking to today. We have a number of different drugs that are available that are well tolerated and in certain populations effective, but we still have a large number of individuals that do not have their blood pressure appropriately or adequately controlled, which has led to an increased interest in understanding what are the molecular basis for the increase in blood pressure. So what you're looking at here, what you're really treating are the symptoms. We know there's an activation of the renin-angiotensin system, and if we can decrease that, we can lower blood pressure. We know there's an increase in volume expansion. If we can get rid of the volume, we can treat that. But what's the underlying cause that's triggering all those things to happen and start in the first place? Immune cells have been implicated really for a pretty long time. There have been studies in the literature going back to the 50s saying, well, maybe there's an immune component, maybe there's an immune component. It was really this paper here published in 2007 by Tom Guzik and Dave Harrison that really put the immune system on the map as far as a potential mechanism to control blood pressure. So what this study did is they took C57 Black 6 mice and RAG1 knockout. The RAG1 knockout lacks B cells and T cells. They treated them with either sham or angiotensin 2 and measured blood pressure. This here is really just represented better over here. So we'll look over here. Systolic blood pressure and diastolic blood pressure. The RAG knockout black bar, hopefully it's as evident and easy to see that the black bar is lower than the red bar. So animals that did not have B cells and T cells had a depressed increase in blood pressure in response to angiotensin 2. They also did this with docosalt. They saw the same thing, a depression in the hypertensive response, two different stimuli if B cells and T cells weren't present. They went on and did adoptive transfer. They put back T cells. There was no change. They put back T cells, and they completely recapitulated the response that was seen in the control animal, really suggesting that T cells were going to be critically important in the development and maintenance of hypertension. Since this time, and that was at this point 2007, which to me doesn't seem like it was that long ago, but to many of you, you're thinking, wow, that was a really long time ago. We've learned a lot. So there has been a lot of ongoing studies. We have learned, you can kind of, in some respects, put anything you want up at the top of this graph, but maybe some of you will like neurohormonal drive. You get the activation of innate immune cells. The activation of these innate immune cells leads to the production of cytokines and other factors, which then drive the recruitment of adaptive immune cells into key organs that are important for the control of blood pressure. We also know that these cytokines themselves do a number of different things to cause changes in blood pressure, vascular tone, vascular function, and other things. So we know a lot, but there was a lot of things that we didn't know. And this is where I kind of started coming into this. So all of the early studies were done in males. When I did our first study, when I think I talked to the students about this, we did a microarray study when I first went to Georgia, and we looked at differential gene expression in mesenteric arteries of male and female spontaneously hypertensive rats. The genes that were most highly differentially expressed were all inflammatory mediators, cytokines and chemokines, and they were all higher in the female than they were in the male. Typically, if you think about autoimmune disorders, or immune-mediated disorders, you think about autoimmunity, and women are more likely to develop autoimmunity and autoimmunity-based diseases than men. So I actually started out my hypothesis thinking, well, maybe immune cells are going to be more important in driving the hypertensive response that you see in females than you do in males. And I was actually at a conference, it was an APS conference in Colorado, and the guy next to me, the poster next to me, was an immunologist at Augusta University. So we went home from that meeting and we started measuring T cells. So are there sex differences in T cells in hypertension? Are there differences in the contribution of T cells to blood pressure control? And are females always protected? This is a loose outline of what we're going to talk about for the next little bit. So the first thing, so yes, I thought this is going to be a nice little foray. We'll do this cute little experiment. We'll go, we'll measure some T cells, we'll find something brilliant, we'll publish it, and we'll move on. But just simple, the sex of the animal impact the T cell profile. And I actually thought that females were going to have more T cells than males. We focused on T cells for a number of different reasons. This here is kind of looking at all of the, and it might not be all, but a number of different immune cells have actually been implicated and suggested to control blood pressure. Most of them, monocyte dendritic cells, a majority of T cells, B cells, and macrophages all tend to promote increases in blood pressure. But we have the relative population down here, the T regulatory cell, has actually been shown pretty consistently to limit increases in blood pressure and decrease the overall hypertensive response. So to start out, we thought we'd look at these T regulatory cells, CD8 positive T cells, and the Th17 cells. Some of the first ones that we looked at. We looked at CD3 and CD4 as well. But just kind of, we picked a cell type to start with just to see what we were going to find. So the studies were done in male and female spontaneously hypertensive rats. The SHR is a model of hypertension that as they age, they have a progressive increase in pressure. And as I think I have on the, yeah, I can jump ahead. This is what blood pressure looks like in young adult male and female SHR. Males are the solid line on the top, females are the line on the bottom. So the studies that we're looking at are about 13 weeks of age. So at that age, both males and females have a higher blood pressure than a normotensive control have, but males have a higher blood pressure than compared to females. We also studied normotensive WKY. Why we did this, it'll become more apparent in a moment. We also looked at 5 and 13-week-old male and female SHR. as well as animals that were either left gonad intact or had their gonads removed. We isolated kidney, aorta, and blood. The data that I'm going to show you today is all from the kidney, although we found the same trends in the aorta as well. The blood tends to be a little bit different. I can talk about that later if you like. And then we did flow cytometry just to look at what the T cells were present and were they different between males and females. So this here is looking at total CD4 positive cells, CD8 positive cells, T regulatory cells, which we are defining as FOXP3 positive cells, and Th17 positive cells, which are the ROR gamma T cells. In all of them, we have the male and female SHR first, with the WKY after them. Males are the solid, females are the open. So if we just look at CD4, start there. First thing that's obvious, in any of the immune cells you look at, the SHR have way more cells in their kidney than the WKY. So the hypertensive rats have more of each of these subtypes than the WKY. Male SHR had more CD4 positive cells than females. Interestingly, and we have not pursued this yet, females actually have more CD8 positive T cells than the male. So the CD8 are often thought of as those cytotoxic cytolytic T cells, but there are regulatory CD8 positive T cells. Based on some of the other things, I think there's a possibility that that could be what we're measuring here. We haven't gone back on to differentiate and look at the CD positive subtypes that are present in the kidney. If we look at T regulatory cells here, our females had more T regulatory cells than the males. Again, the T regulatory cell is anti-hypertensive and suppresses an immune response, and females have a lower blood pressure than the males, whereas the Th17 cell was higher in the male than it was the female, and males have a higher blood pressure. So we at least had a phenotype that would suggest that females had a more anti-inflammatory profile consistent with their low blood pressure. We also have some key cytokines, so 17, and again, males have more than females, and IL-10, which is an anti-inflammatory cytokine released by T regulatory cells. Females had more of that than males did. So across the board, the females had a more anti-inflammatory profile than the males did. But why are they different? So we address this relatively simply. More often than not, people want it to be hormones. So we did the hormone experiment. But the other thing is blood pressure. So at the age that we are studying the animals, there is a sex difference in blood pressure. The fact that the male's having higher blood pressure, could that be part of what's driving the difference that we see in the immune cell profile? So we first did the hormone question. So again, we gonadectomized the animals at 10 weeks of age, isolated kidneys at 13 weeks of age. The intact animals are the first two, and these are the T regulatory cells. Again, we saw that we confirmed the data that we had before, saying the intact female had more T regulatory cells than the intact male. If the Th17 cells, the intact male had more than the intact female. But hormones seem to be anti-inflammatory in both sexes. So regardless of whether it was male or female, there was a decrease in T regulatory cells in males, there was a decrease in females, but the sex difference remained. There was a decrease in Th17 cells there was an increase in Th17 cells in males, there was an increase in females, but the sex difference remained. So while hormones did seem to be anti-inflammatory, regardless if they were male or female sex hormones, they did not appear to be able to account for the sex differences that we saw since they were maintained following gonadectomy. So we then went ahead and looked at 5 and 13-week-old animals. So 5-week-old SHR had yet to go through sexual maturation, and they had yet to develop increases in blood pressure. So it's kind of a quick way to say, okay, we don't think it's going to be the hormones, but what do we see in young animals? And these are the T regulatory cells, and it's 5 weeks of age. There's no difference between males and females in either T regulatory cells or Th17 cells. With the development of hypertension and the sexual maturation, really there's an increase in females in the T regs, and then robust increase in the Th17 cells. We started to think maybe it was hypertension. So yes, we had studied the WKY and we didn't see a large number of cells there, but the WKY is actually a distinct genotype from the SHR. Whether or not they're a good control or a bad control, I'm not really speaking to, but they're a different genetic strain. So what would happen if we took SHR and just lowered their blood pressure? Do we see these same differences? So we took two different approaches. One, we treated animals starting at 6 weeks of age. So before they had an increase in pressure and before there was a sex difference in blood pressure. Blood pressure was monitored in these weekly by tail cuff. In my laboratory, SHR, especially female SHR, do not survive telemetry implantation before about 8 to 9 weeks of age. They're simply too small to survive the surgery. So we measured by tail cuff in this study. So these are the control males, these are the control females. We treated the animals with a combination of hydrochlorothiazide and reserpine, and we were able to blunt the increase in pressure and abolish the sex difference in blood pressure. So this was preventing the development of hypertension. We also said, well, what if you reverse established hypertension? So we took animals that were 11 weeks old. So here there are at 11 weeks of age, here's the males, here's the females. This is done with telemetry, and then treated them acutely for 2 weeks to, again, abolish the hypertension and get rid of the sex difference in blood pressure. So what happens to T regulatory cells in particular is what we were interested in, but the Th17 cells as well when we do this. So these are the animals that were treated for 6 weeks. So here's our vehicle treated male, and there's our blood pressure lowered male. As you can see, there's no change in the males in the number of T regulatory cells, but in the females, if blood pressure does not increase, T regulatory cells do not elevate. There was really no difference in Th17 cells. There was no significant difference. It might look like it's a decrease, but it didn't reach statistical significance, and there was no change in the females. So we saw a blood pressure dependent increase in the females. Similarly, if we treated them acutely for 2 weeks, so if we lowered blood pressure, no change in the T regulatory cells in the male, but in our females, we saw a decrease in the number of T regulatory cells. So we thought maybe that the T regulatory cell was then going to be critically important in the female, and they had this compensatory rise in T regs in order to limit increases in blood pressure and protect them from the negative cardiovascular consequences of increases in blood pressure, which is exactly what I say on my summary slide right here. So males have a more pro-inflammatory and females have this compensatory increase. But does it matter? So all we're looking at here is I'm telling you this is what's present in the kidney. This is what's present. Does it actually mean anything for blood pressure control? So our hypothesis 2 was that T cells were actually going to contribute to the sex differences that we see in blood pressure. We started out thinking maybe we would do this in SHR. So again, we took 6 weeks old. This is just female SHR, and we treated them with anti-CD25. So IL-2 binds to CD25 on T regulatory cells. That activates the activation and proliferation of the T regulatory cells. So blocking CD25 has been shown in a number of different studies to decrease the number of activated T regulatory cells. So we took a subset of females to measure blood pressure weekly, treated them, and it kind of looks like maybe if you treat them with anti-CD25, the increase in pressure, which is what you would have hypothesized. But to me, it wasn't a great outcome. And to be perfectly honest, the cost of treating them with the neutralizing antibody for this period of time kind of was prohibitive to doing a larger study. At the same time, I had a new graduate student starting in the lab, and she was interested in Dokosol hypertension. As I mentioned before, Dokosol hypertension had also been shown in that paper by Guzik and Harrison to be dependent on T cells. We know Dokosol hypertension also has sex differences, so we decided to switch to the Dokosol model of hypertension to ask this question more completely. So we take Sprague-Dawley rats, 12-week-old Sprague-Dawley rats. All rats get a unit nephrectomy. Half the rats get a Doka pellet and saline to drink. The other half get no pellet. We do a sham surgery, and they get tap water to drink. We measure blood pressure via telemetry and measured T cells. We also had a subset where we treated with Doka or Doka with anti-CD25 to actually assess the contribution of CD25 to Dokosol-induced increases in blood pressure. So this is just the control, males and females. So this is the initiation of Dokosol treatment. You see a robust increase in blood pressure really quickly following the start of the treatment, and males have a greater increase in blood pressure than our females. We went on in these animals and measured the T cells, And again, I was expecting we would see the same thing that we had seen with the SHR, because we also saw that same thing with the Anch2 models, and we had seen it with some other things. The DOCA ended up being a little bit different. So here are our CD3 positive cells. In males and females, our uninephrectomized controls and following DOCA salt hypertension. So effective treatment was significant. So DOCA salt hypertension increased CD3 positive cells, but the increase was comparable in males and females. Here's looking at CD4 positive cells. Again, there's an effective treatment. So DOCA salt increased CD4 positive cells, but males had more than females. Here's our T regulatory cells. So our UNX controls, so female Sprague-Dawley rats have more T regulatory cells than male Sprague-Dawley rats. DOCA salt did nothing to T regulatory cells in the males. They tended to decrease them in the female, but even these had more T regs than males, maybe. So it was like, okay, this isn't not what I wanted to see. I wanted to see this go up, but I was going to roll with it. We can work with this. And then the TH17 cells, there was an effective treatment. So TH17 cells went up, and males had more than females. So we did have a sex difference in T regulatory cells. So we went ahead and repeated the experiment using DOCA plus an anti-IgG or DOCA plus anti-CD25. We confirmed our ability to decrease T regulatory cells. So this is in blood and this is in the kidney. We did see significant decreases with treatment in both sexes. And this is our blood pressure response. So the top is all 4 groups together, but I broke them apart because I think it's a little bit easier to look at. Here's the males. I think you'll agree with me, treating with anti-CD25 really didn't do anything to the DOCA-induced increase in blood pressure in the males. Whereas in the females, these are our DOCA alone. These are DOCA with anti-CD25. We significantly increased the blood pressure response such that the females pretty much are superimposed with the males. So treatment with anti-CD25 abolished the protection against DOCA salt hypertension that we saw in the female, causing us to conclude that females are more dependent on Tregs to control their blood pressure versus their male counterparts. I'm going to now switch gears. I will tell you we're kind of continuing in that vein. So the ongoing studies from that are, well, why do females have more? And is it production or recruitment? So we've looked at splenic T regulatory cells. They're the same between males and females. So we don't think it's production. We think it's a recruitment effect. So how is it that females are actively recruiting more T regulatory cells than males? I don't think it's hormone mediated. The hypothesis we're looking at right now is a nitric oxide dependent phenomenon with IL-33. Happy to talk about it, but I want to move on. So here we had a case where the females were protected and they have increased in immune cells. This here is looking at hypertension awareness, treatment, and control in non-Hispanic white males and non-Hispanic white females. So in each of these, the males are highlighted in blue, the females in red, non-Hispanic Black males and non-Hispanic Black females, and Mexican-American males and Mexican-American females. You can try to look at all the numbers. I'll tell you right now, in each case, females are more aware of their hypertension. They are more likely to be treated for their hypertension. They are more likely to have their hypertension under control. But if you look at the adjusted hazard ratio of cardiovascular mortality associated with hypertension, it is significantly higher in women. It's 2.356 versus men, 1.937. So eliminating hypertension has been thought it can reduce cardiovascular mortality 30% in men and 38% in women. So women are dying from hypertension more often than men, despite all of these other things, which up here would suggest to you that women are going to be better off and they're going to be protective. So why might that be? Anybody want to, I've been talking for a while. Anybody think they have an idea why? Okay, fine. I will continue to talk. I thought I'd hand the microphone over. So we started looking at fat. So chronic consumption of a diet high in saturated fat has been known for a very long time to promote the development of cardiovascular disease. as a general practice, we hear, and maybe it's not this department because y'all look very healthy and appropriate. Most people consume way more fat than they're supposed to. So the average is less than 10% daily intake. 75% of Americans consume way more than that. And, you know, I'm not going to say that I'm average, but I'm certainly not consuming what I should. And high fat diet promotes increases in adiposity, blood pressure, and inflammation. We also know that women are more susceptible to the cardiovascular complications associated with obesity than men. So an increase in 5% energy from fat in women increases risk of coronary heart disease with no relationship in men. Women are more likely to be obese than men, and being overweight accounts for 62% of hypertension cases in women versus 34% of cases in men. More deaths are attributed to high BMI in women versus men. So we have a lot of overweight and obese women eating more saturated fat than they should, which promotes increases in the development of cardiovascular disease. So understanding how a high-fat diet is doing this could be critically important to saving the lives of women, especially if you look through, you know, I live in that belt in the South where we have a lot of obesity, a lot of cardiovascular disease, and a lot of stroke. Sorry, there was too many clicks there. So we wanted to know if there was an animal model that we could study this increased susceptibility of women to high-fat diet. We had done a study a few years ago in Dahl salt-sensitive rats. So this here is a study that we published. Well, I don't know where it's up here someplace. So Leah Taylor. We took 12-week-old male and female Dahl salt-sensitive rats, put them in a normal fat diet or a high-fat diet. The high-fat diet was 59% kcals from saturated fat. Bioserve, the control diet, was the normal fat from Bioserve as well. We treated them just for 4 weeks. So I'm certain all of you that do high-fat diet studies, 4 weeks is not very much time. But we saw increases in blood pressure. So this is systolic blood pressure in males on a normal fat diet, males on a high-fat diet. We then repeated it in, or I guess this is just over time. This is tail cuff as well, and percent increase in pressure. So we saw increases in pressure in both males and females. They tended to be pretty much the same. So the blood pressure in males and females was comparable. End of the treatment. This here is work out of the University of Michigan. They're also studying salt-sensitive rat placed on a high-fat diet. They do a 24-week high-fat diet. They've got their body weights here, mean arterial pressures here, though. So increase in pressure. These here are our control fat diets. These are our high-fat diets. You cannot differentiate the males from the females. So we have comparable increases in blood pressure in males and females to a high-fat diet. If this rat model is put on salt, you also see increases in blood pressure, but the males always have a higher pressure than the females. So this is an 8%, and 8% is a pretty, you know, it's a sledgehammer. Males have a higher blood pressure. Both of these are 4% high salt diet, and males have significantly more pronounced increase in pressure to salt in all of these cases. So in my thinking, if the females are typically protected, the fact that we don't see that with high fat could indicate enhanced susceptibility. Here is not. This is just salt. So our next hypothesis was that female Dahl rats would have greater increases in blood pressure as well as inflammation when placed on a high-fat diet when compared to their male counterparts. So we took 5-week-old male and female Dahl rats. At 5 weeks of age, they were randomized to the normal fat or high-fat diet. We measured blood pressure via telemetry from 11 to 16 weeks of age. At the end of the study, we collected the kidneys and we looked at these same immune cells that we've been talking about throughout this entire time. Here's what we found. So our control, so these are slides from Lindsay Ramirez. She's a PhD student who graduated. She's now doing her postdoc in University of Chicago. She likes pink. So what I would have had as red is now okay. Control fat fed female here. These are control fat fed male here. High fat fed males, there's a significant increase in blood pressure in high fat diet males. As you can see, a much more pronounced increase in pressure. The one thing you'll note is, I mean, this is not a small n. I mean, we have quite a few animals in here, and to have variability like this with telemetry is a little bit unusual. In our females, we have responders and non-responders. So within each group that we've done, there have been some that have had pronounced increases in pressure, some that have moderate increases in pressure, and some that don't have much at all. I find that to be wildly fascinating. It's something that we probably need to continue studying, but regardless, it does suggest that females were more susceptible to the high fat induced hypertension than the males were. This isn't because of huge differences in body weight gain. So we measured body weight weekly. There was no real difference in weight gain in normal fat fed rats and high fat fed rats. They both tended to have similar body weights. Males were heavier than females, but the normal fat, high fat fed of the same sex tended to be comparable. However, when we measured perirenal adipose tissue and gonadal adipose tissue, there were significant increases. So we were definitively increasing adiposity. And I'll show you some NMR data in a little while. So the difference wasn't because of body weight, but we did have increases in adiposity. These are the immune cells. So here are CD3 positive, CD4 positive, Th17 and T regulatory cell. There were no significant changes in total CD3 positive cells or CD4 positive cells. There looks like there could be trends to have them increase. This is in the kidney, but we did not see significant changes there. However, in the subsets, so the Th17 cells, which are the pro-hypertensive, pro-inflammatory cells, tended to increase, and they increased comparably in both sexes, and T regulatory cells decreased, and they tended to decrease the same, although we went on in a subset of animals, and you can tell now this is one of my slides versus Lindsay's slides because we're now back to red versus pink, but that's okay. We looked in the aorta, the kidney, and adipose tissue and looked at the ratio of Th17 cells to T regulatory cells. And it actually does suggest in response to a high fat diet, females have a more robust increase in the overall pro-inflammatory profile across the board in each one of these different organs. So we know that inflammation is common in obesity, and a number of different things have been studied. A lot of it has focused on macrophages, cytokines over here, and we'll come to the NLRP3 inflammasome in a bit. But what about the role of the T cell? So we wanted to directly test that. We wanted to test whether or not T cells contribute to high fat diet induced hypertension. We were able to do this by using a CD247 knockout Dahl rat. These rats were developed in the Medical College of Wisconsin for Dave Matson's laboratory. And these are just showing these cells. So there's total PBNC, CD3, CD5, and CD45 positive cells in the sham control or the genetic control or the knockout. There's a pronounced loss of our CD3 positive cells. This is in response to salt. When they take CD47, you have a depressed response to salt. So this is the animal model that we decided to use. We took 5-week-old male and female Dahl rats and CD247 knockout rats, put them on the high fat diet, and looked at our blood pressure response. And this is what we found. These are separated by sex because I wasn't interested in having 12 lines on a graph and trying to get into 3- and 4-way ANOVAs. So these are controls on a normal fat diet. Oh, let me get this right. No, the dark pink are our controls. The light pink are the CD247 knockout. So here are the animals that we showed previously. Oh, there's a couple more N's in here, a couple different N's in here. We had that pronounced increase. Here are the CD247 knockouts. Here are the 247 knockouts on high fat. So not having T cells completely abolished that increase in blood pressure, although maybe there's a small increase, but you certainly don't see this robust increase. And we saw same thing in the males. So the light blue are the CD247 knockouts. And then again, you don't see the increase in pressure that we saw in the other ones, suggesting that having T cells present was important for fat to be able to cause an increase in blood pressure. What those T cells are doing, I don't know yet. We did see that there was a slight change in weight gain, but really the difference, so here's everybody here on the high fat diet in the females, the control and the CD247 knockouts in both control and high fat diet tend to be pretty superimposable. In the males, they spread out a little bit more. And what it really is these are the CD247 knockout on normal fat diet. They don't grow as fast. So they tend to be smaller. If you actually look at the delta, the change in weight gain in the knockout could actually be greater than the change in weight in the control animal, but they're not robust differences in what this means. I don't quite know yet. It's not because they're eating less fat. So we measured fat consumption. It goes up in both, and it goes up similarly regardless of genotype. I didn't know the slide was going to be here, but that's a good thing. So at this point, I think we have a model that we can actually study. So it reflects what we see clinically. We have an enhanced susceptibility in the female relative to the males. We find that high fat diet does indeed induce inflammation, and T cells are contributing to both the metabolic and the physiological phenotype that we're seeing in response to high fat diet. The last two things I'll talk about, I have no idea where I have time. I think I can get it done. Are how are the T cells being activated? What about salt versus fat? So to come back to that question that you asked me before. So this comes back to NLRP3. So the NLRP3 inflammasome has been shown to contribute to diet-induced weight gain. Free fatty acids bind and activate NLRP3. NLRP3 then induces the activation and expression of IL-1 alpha or IL-1 beta and IL-18, which then can drive T cell activation and T cell recruitment. We measured NLRP3 in adipose tissue, peritoneal macrophages, and renal macrophages of male and female Dahl rats on a normal fat or high fat diet. And in each case, we saw the greatest increases in NLRP3 were in the females, regardless of the organ in which we measured it. We also measured IL-1 beta, and females had more circulating IL-1 beta, greater IL-1 beta expression in macrophages in both the peritoneum as well as in the kidney. Well, why would that be? So as I said, free fatty acids can activate NLRP3. And what do we know about free fatty acids following a high fat diet in males versus females? I'll tell you, we do not know a lot, and we have started to look at this in collaboration with Sean Campagna at the University of Tennessee. He did a quick measure of free fatty acids, circulating free fatty acids in males and females, did find there were differences. These were the top that were differentially expressed. All were higher in females when compared to males, and each one of these has been shown to be linked to increases in blood pressure. So could it be differences in the gut microbiome, differentially metabolizing the high fat diet, have different free fatty acids, differential free fatty acid production in males versus females, could be causing greater NLRP3 activation in females, then subsequently causing that greater increase that we see in T cells and promoting differences in adipose tissue expansion. So that's kind of the direction that we're going in that study. The last question is one that is real. So I showed you this before. So the salt. Males have a higher increase in pressure. Males have a higher increase in pressure. Males have a higher increase in pressure. I just showed you don't see the same thing with fat. And if you look clinically, women are more likely to have salt-sensitive hypertension than men, yet one of the primary animal models that we use to study salt sensitivity shows that the females are protected. But how often do we eat fat without our good friend salt? Not very often. So is the fact that the females on high salt have a lower blood pressure because we're limiting their fat intake? I'm going to tell you, I think maybe. So I'm doing this study in collaboration with Justina Baez-Batad, who's an assistant research scientist in David Matson's group. We took male and female Dahl salt sensitive rats. Females are in blue, males are in red. Put them on a normal chow. Put them on high fat. So we high fat loaded them for 2 weeks. here, and then we put them on high fat, high salt. So the same BioServe diet that was the high fat, we then just added the salt into the same diet. So the normal chow is not, but these two diets are basically the same except for the salt content. And when we put these animals on high fat, high salt, your females tended to have a greater increase in pressure. Now, this is preliminary data, and I'll tell you, it looks a little weird to me as well. I'm not really sure what this drop was about, but if nothing else, the blood pressure is higher, which would suggest to me that if we actually put them on the high fat, high salt together, it's going to be the fat is potentiating that response to salt, which is going to be more indicative of what we see clinically and maybe more relevant if we want to learn better what's happening in response to a Western diet. So again, I think if we can understand how the high fat diet is differentially impacting the males versus the females, we could have a significant impact on a number of different cardiovascular parameters in a number of different individuals. So I started with the question, do T cells underlie sex differences in blood pressure control? I think maybe they could at least be an important part of the equation. And the fact that regulatory cell in particular seems to be important to me is very intriguing. Well, we're not going to start giving broad acting immunosuppressants to people on hypertension. You can specifically target T regulatory cells. They're giving autologous T regulatory cells for other types of conditions. But more importantly, if we can figure out the pathway by which females are having greater increases in T regulatory cells, are there key spots in inflammatory cascade in general that we could target to get selective changes in key parts? So here, these are just 3 different examples. This is out of Catherine Sandberg's laboratory, although Hedwin Brooks has done similar things, looking at the RAG knockout mouse. In response to angiotensin II, males almost always uniformly in multiple species, whether it's mice, rats, dogs, pigs, humans, males have greater increases in blood pressure. In the RAG1 knockout, that response tends to be completely abolished. So if you don't have T cells, you don't have the sex difference. This is Casey's work showing that the T regulatory cell is critical here. And here we see the difference in T size. And as I showed you, they don't have cells. You don't see those increases in pressure. You don't see the cell. I having greater understanding of how the immune system is differentially regulated in males versus females could provide us some insight and some novel ways to treat blood pressure and lower the overall burden of hypertension. So with that, I did absolutely none of this work. This here is Lindsay Ramirez. She is an outstanding student. She did most of the high fat diet stuff. Casey Belanger is back here. She was the person that did the dope salt stuff. Riaz Muhammad is an assistant research scientist in the group. He did the anti-CD25 in females. These 2 are just my children, so they're obligatory. I think those are the key people that did. Lizzie right here also did some of the high fat diet stuff. So huge thanks to them. I have outstanding collaborators, and I'm happy to take any of your questions.
Showed that females, once they get to the 75+ range, they have increased prevalence of hypertension. Would you suggest that it's the ratio of the T regulatory cells to other T cells, or is it that the T regulatory cells are decreasing with age?
Great question, and it's probably a little bit of both. There's not a whole lot that's been done with aging per se. Brooks has published biology that they see in those. I don't know exactly. And I think also probably ask me. I'm aging. Yes, I can do that. The question I was just answering is, do you think that the increase in hypertension in old age is in part due to a loss of T regulatory cells.
I had a question about so that it pays for individuals with autoimmune
So the question is, in autoimmunity, is there an increase in the development of hypertension or risk of hypertension? The answer is in most cases, yes. So, I mean, lupus nephritis, there's a huge increase. For arthritis, there's an increase. In all autoimmune diseases, I don't know. In many, yes. Sure. And they've actually shown it in clinical populations where they treat individuals, I think it was with rheumatoid arthritis, with mycophenolate mofetil, which is a broad-acting immunosuppressant. There's a decrease in blood pressure in those individuals. They take them off the MMF and their blood pressure goes back up. So this was Barry's defense. This is Barry. The first meeting I took Barry to, my good friend Dr. Gillis giving her talk, and and there's no Barry. So I text him, Barry, where are you? Oh, I met some people from Cincinnati and I've gone to the coast to see sea lions. This is Barry in his suit surrounded by sea lions. So I had everyone in the group, and I made them Barry shirts, and at his defense party, we all proudly displayed Barry.
Yeah, blood pressure response, males and females in other models of diabetes, obesity, for example, like high sugar, high glucose diet. So I've seen difference and data from our lab. We've shown that the response appears to be more similar to either the salt or the age, but males are more susceptible.
Yeah.
So
So no, we have not looked at. So Connor, that's all over here. He is doing some stuff now, looking at fructose feeding. So I was thinking about he's just doing males. So I figured we could start a little bit there and measure some of these same things, but I don't know, and I don't have a good answer for you on that.
Most people in the United States, they get their sodium not from salt in their food, but from salt additives. And so what do you think, how do you think, if you think that would affect it at all?
So the question, sorry, I'm not sure I repeated that question. So would the response that we see with salt be the same if instead of using sodium chloride, we use a salt additive? Yeah, I don't know. So good question. Not one that I have a response for. So what would you suggest we try? They're rats. They eat their own poop. They'll eat anything. That's true. And this would make everything better.
I think my opinion certainly much like translatable to American population.
Sure. So excellent point, and the one we should probably look at. A more translationally relevant form of salt in the diet. You are absolutely loud one. Maybe I'll answer that one. Let's see.
Great understanding of staff. And so my question is thinking about the Mediterranean kind of diet where it is not low fat, but it's mostly monounsaturated. Cardiovascular risk, but they also don't have a lot of salt in their diet or sodium unless they go to the coast.
It is lard. So the question is, what is it? How would you translate this to Mediterranean diet, which is not low fat, but it is not polysaturated? Yeah. So no, this is lard. Lard. You gotta say it like that. My guess is it would be different if we had monounsaturated as opposed to what we're using. I think there's actually a lot of potential cool things in there. So I mean, the salt sensitive fat they're sensitive to everything. I mean, the source of protein makes a difference. It's not based. So looking at that would definitively be something that we're interested in doing. We're working on getting this funded, and then I've got a whole bunch of things I'd like to do, but that's that. How are they doing that? What are they doing? They're my answer. They're activated. They're putting out a whole bunch of IL-10, and IL-10 can cause vasodilation. IL-10 can cause a bunch of things. IL-10 promote. I think it's IL-10. Are there any medications that use IL-10 as a driver? Not that I know of, but it doesn't mean that there are not. Yeah, so the question is, would T cells be different even prior to sexual maturation? So is there a chromosomal effect as well? I would imagine that there is. I would be surprised if there wasn't. I don't know in the 4-core genotype model, which the chromosome effect from the hormonal effect. I'm not aware of studies that have looked at immune cells. It could just be I haven't been in the literature recently enough. And I think they're working on making a 4-core rat. I'm told. I don't study mice, they're too little. Now, a question about the models. You said that you were using SHR and she was really. Could you describe how they are different and what would make you, like what made her interested? Sure. So why would you be interested in one hypertensive model versus another? So a lot of what we've done has been the spontaneously hypertensive rat. The reason I like that model is because it's a model of essential hypertension. As they age, their blood pressure goes up. It goes up in a non-specific way. Blood pressure responds to the same types of drugs that we give patients. If you give them a RAS inhibitor, blood pressure goes down. You put them on a diuretic, blood pressure goes down. So it's kind of thought of as that model of essential hypertension, but it takes time. So if you want to study something, so when we're doing those 6-week interventions, if you're going to give a drug that costs $300 every time you inject the rat, it can become prohibitively expensive. So when Casey was in the lab, she had an interest in, actually she was a neuroscientist background, and she wanted to look at some brain chemistry, and for some reason she came on a paper showing that the Docetxel had something that she was interested in. I don't know exactly what triggered it. But in her mind, it was a more translational model because she said, well, here you've got the neurogenic component, you've got the salt, you've got the vasodilation, you've got the genic component. So those appealed to her, and that's why she wanted to study that model. I was perfectly content to do that because it also took us from 6-week treatment to week treatment, but we still had the sex difference in blood pressure. So I have a student now who's following up, and he wants to do angiotensin 2 hypertension. As he says, well, we're using all these RAS inhibitors, so let's use an angiotensin-dependent model. So he's kind of following up with some of that early stuff, looking at T cell activation. For me, it doesn't really bother me looking at different models, because if you look at multiple different models and you tend to find the same basic trends, that reinforces the fact that we're actually studying something that might actually be important versus some artifact in one rat model. I don't really want to think at the end of my career I've made rats healthier. You like to think that you're actually studying something that will have the potential to actually impact human health. So that helps. Maybe. Yes, please. So the question is in pregnancy, what's happening to T regulatory cells? Yeah, there's actually quite a bit of literature on that. So T regulatory cells normally expand in pregnancy. That expansion of T regulatory cells helps you not the fetus and also helps some of the vascular modeling. The decrease in T reg expansion in pregnancy. So preeclampsia is associated with a decrease in the number of T regulatory cells.
One more question just for clarification. In the large clinical data, you said so the females are more likely to be controlled, have their hypertension controlled, but even when it's controlled, they're still at a greater risk of cardiovascular related death.
Yeah.
So I'm just curious. In my mind, I was thinking perhaps females are developing hypertension, but the drugs that we're using are currently not targeting the mechanisms that are beneficial. But it seems like it is. So those things would say if you lower the pressure with whatever drugs are available, you're okay. That align with, or
So I get your question. So the question is, if I said that women are more likely to get diagnosed, more likely to take their drugs, and more likely to have their blood pressure controlled, why in the heck fire are they dying at a higher rate? My question would be to you, do we even know what optimal pressure is in a woman? Because we have said it has to be, 130/80. The sprint trial did not have enough women in it, and we terminated it early because of the benefits in men. So the sprint trial, you can't even draw conclusions about what an optimal pressure would have been in women. So I would suggest that, and those data are out there in piecemeal, like actually if you lower blood pressure more in women, there are larger benefits. So I don't know that we're diagnosing women well enough or lowering their blood pressure enough to show a benefit. So if we said, okay, in women, it's 120/70, would we not see what we're seeing there?
that's so that control may be controlled for a male for nothing.
So yeah, so the burden of blood pressure on the system of women versus the burden of blood pressure on the system of men, why is it the same? Do we know that that's appropriate? And I don't think we do. That would be my answer, but that's just me spitballing. Yeah.
Just the last optimal blood pressure for men and women do meet males and females effectively, or individuals undergo hormone replacement therapy, such that their blood pressure, optimal blood pressure, change to the other.
So when I say male and female, am I encompassing all genders? I do not know. And exactly. So if you start to look, and there are some data out there, and actually there was more than there is now. I mean, I think there's some more hesitancy in the transgender population now to engage in some of those studies, but I don't definitively know. I mean, I know there are some studies out there from a number of years ago showing, beneficial effects of estradiol treatment in transgender women, but I don't know, and I'm not going to speak to that. I don't know how the hormones and the chromosomes would come together to define what your blood pressure would be, and it's probably not going to be simple.
Okay, well, thank you so much. I think we've over time, but